So I want to thank you for inviting me to give this and to keep me to the end of the session and have me speak right after Mike, which is never easy. And also being a Dana-Farber, I'm very proud to be on the MIT side in this particular case. So this is something really good. I'm not sure how, should I, I'll do it myself. I think we're good. Thank you. I think. We'll see. Yeah? Are we good? All right. So, thank you very much. So basically, Mike gave you a really great introduction. I don't know what he's talking about. I mean, the problem is I've never seen pornography, so I don't know exactly what he meant. No, I'm just kidding. So, but if you're... Maybe he did. I don't know. Anyway, but I've heard that one before. It's a funny one. And so, if you want to have a little bit more information, that's interesting. So that slide is not. Let me stop one second. We had that problem a little bit earlier. I'm not sure if the. Can you see it? Okay. Sounds good. Sorry. Here we go. I just want to point you to a review, a set of reviews that were okay. So now I'm losing everything. Can you guys look in this direction? Do you see? Yeah? Is that right? Is there somebody to help? We had somebody, so we'll go. All right. So, and the fact is that, yes, Mike told you a lot about measuring and modeling things like apoptosis or other pathways in cancer. We're sort of interested, and there are many other approaches here, and if you don't know much about systems biology, you want to learn about it, I think that special issue of Cell is really great. We wrote one article with Laszlo Barabashi, a physicist with whom I've been working for the last 6-7 years on interactome networks and human disease. So to put this in a framework, what Mike told you about is a lot of logical relationship stuff, dynamic information flow, and this is extremely important. That's actually really the end point of systems biology. However, to build these models, you need at some point or another to better characterize the physical interactions between things. That's sort of the starting point. And I think if we work together, which we actually do quite a bit, in addition to, by the way, imaging cells also is very important, all together we'll be able to better understand the systems that go wrong in cancer and in other diseases. So let me define the interactome networks a little bit better here. So those are basically macromolecular interactions. They contain protein-protein, DNA-protein. Metabolic interactions as well can be part of the interactome networks, depending exactly on how you define things, as well as non-coding RNAs and their targets. And we're pretty much specialized on protein-protein interactions, but we're interested, of course, in integrating all these different networks. Maybe what you don't know is what we do is we simplify the beauty of the structures of proteins and their logical and functional relationships between each other, and we basically summarize things by little nodes representing proteins or macromolecules of different kinds, and edges or lines between them that represent biophysical interactions. And the main hypothesis of our work is that if we were to be able to map these interactome networks at the scale of the whole genome or the whole proteome, that there might be properties in these networks that relate to biology. So to give you a little more flavor of what's in that review that I was pointing you to a second ago, the idea is that Mendelian disorders, complex traits, and of course, as we're interested in tonight, tumors and cancer in general, might be better understood if we could combine genotypic information. We'll talk about cancer genomes in a second. The knowledge of Mendelian mutations, GWAS, $2 billion having been spent in the last 10 years or so, and potentially cancer-associated genes, if we were to study the systems and the networks that are in between the genotype and the phenotype. That's the whole idea. And again, Mike did an amazing job at showing you examples of that. A little bit of, just prepare this for this particular audience. Of course, the interactome field is still in its infancy. For example, in 2011, there were 100 times more papers published with genome as one of the topics than there were about papers with the interactome word as one of the topics. So we're really still a very small field. That might be why you haven't heard of us quite yet, if you haven't. But still, we're growing, I'm sorry, and we're growing relatively fast. So for example, if you look at the fold increase in number of publications since 2005, where we really started publishing some of these network maps that I'll talk about, well, actually, we've been growing fivefold. The genome really has been growing 1.5-fold. So it's sort of an active field here. We're looking towards the future, not the past. The other thing that really contrasts the genome and the interactome is that mutations, lists of mutations, cancer genomes, basically give you a list of wrong phone numbers if you were looking at the phone book. We're not doing phone book stuff. We're doing Facebook stuff. We're doing interactions. We're trying to understand, if you were interested in sociology, we're trying to understand who friends whom and who defriends whom in the cellular networks. Okay? Talk about Facebook. It's unbelievable, right? When I started this in 1993, I mean, I barely knew what the internet was, right? Seriously, I was in Ed Harlow's lab, and the idea was, well, one day we might actually, we'll have the genome sequence, we'll put all the proteins together, I'll show you in a second, and we'll have a network. But now, we're not there yet, but we do have networks. And so physicists and engineers and other people and computational biologists can actually look at these networks and look at their properties. And I can tell you that these properties are absolutely not boring. We're all pretty much on Facebook. We're nodes in the network, and we define edges between us by friending and defriending each other. So what do? We map protein-protein interactome networks, we model the properties of these networks, and then, and that's most important for this crowd today, we use some of these properties of interactome networks to better understand cancer. So what I want to do is to give you just a couple of vignettes in the time that I have left. So first of all, it can sound a little boring. It's not really rocket science. Imagine the proteome in front. If you can close your eyes and see 20,000 proteins, for example. I'm excluding isoforms for those who know about splice variants for now. You have 20,000 by 20,000 combinations. That's 200 million pairs that can be tested in someone's lifetime. We're not quite there yet, but we're making really good progress. It's enormous. Basically, the idea is you can measure whether or not two proteins can interact with each other. We use a panel of assays to do that. We've published many, many papers about the quality of these data sets, the sensitivity, the specificity, and as many of the speakers before, I don't really have time to go through the details. Suffice it to say is that we end up with networks that pretty much look like the Facebook network. Okay? And that's sort of the unperturbed network that we've been measuring for many years. The latest set of data sets that we've produced, we actually haven't published yet, but it's on the internet already. That's also something about systems biology, is we tend to really share a lot of stuff with the community, right, Mike? I think he's doing this with his, I paid him $20 to do that. But the bottom line is this is not published yet, and it's an enormous data set. Let me show you why I say this. I'm measuring things here at equal quality of protein-protein interaction signals. The blue is showing you what happened in the literature since, P53 and large T or RB and large T were discovered, and how protein-protein interactions have increased in the literature. Red is our work, 2005, Nature paper, first map, and then it took us a while to basically redesign things and go faster and reduce the cost, etc. The data set that we're about to publish is up here. Okay, so we just, as happened in the late 90s or so, we're in a stage now where we're basically going faster at much lower cost, and in many ways with higher quality, we're going faster at mapping this huge beast, which is the interactome networks, than the whole of the community. Okay, so properties, one slide. Again, we don't have enough time, but just think of that when you go home, that there's an entire field of people, physicists, again, engineers, computational people, computer science, who are really interested in the properties of these networks. And you might think, what am I going to do with a bunch of nodes and edges between them? It's really fascinating. These things have absolutely gorgeous properties, all the way from sociological networks, Facebook, down to the interactome networks that we've been able to map so far. How do we use this for cancer? And that I'm subdividing in 3 sub-vignettes now. We can use this to help finding new cancer genes. We can help contextualizing cancer genes. I'll show you what I mean by that. Close your eyes, think about a map, and that little arrow, you're here. That's the sort of thing we do. And then finally, I'll give you an example or two on functionalizing novel cancer genes. Okay, find new cancer genes, just 1 or 2 slides. This was an example of a collaboration that we had with David Livingston and many others. Miguel Pujana, when he was in my lab, decided to build an interactome network model seeded by 4 known genes or gene products involved in breast cancer susceptibility: BRCA1, ATM, CHEK2, and BRCA2. Again, I don't have time to tell you how he built the networks, but basically basically, again, measuring the quality, the sensitivity, the specificity of these things. And then importantly, he then ranked the likelihood that the nodes in this network would indeed be involved in breast cancer susceptibility. And you might think that this is not right because it's just the top of all of them. The arrow is very small on that side. I apologize. But it turns out the top ranking node was called HMMR, involved in centrosome dysfunction. It turns out David Livingston since then has proven all the biochemistry of this. And it turns out Miguel in his own lab has gone back and basically used human genetics, GWAS, etc., to demonstrate that HMMR is a very likely modifier of the penetrance of BRCA1. Now, you might say, well, okay, I've heard that story before, but I can tell you from just the genetic data, it would have been very hard to infer that. So it's a very good example of basically using and combining the power of interactome networks around seed nodes that you know something about, and then push that all the way to the genetics. Contextualizing things. Mike told you, I mean, you know, drugs, he was using pairs of drugs, using the model to contextualize. That's what it is. Look at the analysis, one of the analysis we did with Laszlo Barabashi. It turns out if you go to OMIM, the Online Mendelian Inheritance in Man database, started by Victor Marcusic in the 50s, you'll find 3,000 or something like that genes involved in any one or more genetic disorder. You'll find a great number of genes involved in that as well. And so it turns out there's a bipartite graph that links the genes involved in diseases and the diseases that can occur if you have mutations in any one of these genes. And it turns out there's nothing simple. The old concept, one gene, one mutation, one phenotype, one protein, is really behind us at this stage. It was great for a while to let us, you know, really develop the molecular biology concepts, but it's gone now. I mean, really, it's dead. Look at the network that we get out of this. This is a network of diseases. In this case, the nodes are diseases, okay? And I link them with each other. I put edges between them if they share at least one gene that mutations in which will increase the susceptibility to that disease. This is the cancer corner, for example. 2 observation. Many of the cancers are linked with each other. Nothing is in isolation. You show this to the development office, and it's very hard to say that you really want to work just on prostate cancer, for example, right? Because everything is so interconnected. In addition to that, the cancer genes are connected to the rest of the network as well. So even cancer cannot be really thought of as an island in this particular type of model. Functionalizing, my last example, if you don't mind. Again, I mean, there's not a Nature issue now that doesn't have a cancer genome paper, right? Which is wonderful. I mean, this field is exploding. By the way, a lot of that was started here, personalized here in Boston, Dana-Farber and MGH. And it's wonderful. I mean, in 5 to 10 years, we will have a great idea. There will be an atlas of what the cancer genome looks like. All the tumors, all the genes mutated, etc. But the problem is still you have the wrong phone numbers when you do that. You still want to see how the Facebook rewiring occurs. Mike actually used the word rewiring quite a bit. So I'll give you just one example of something we did in that context. That's a Nature paper that just came out a couple of months ago. What we were trying to do here is interpreting cancer genomes using systematic host perturbations by tumor virus proteins. So what we did here is considering interactome network models that we had, what we wanted to do was to the extent to which DNA tumor virus proteins actually touch the host and rewire the network, and the extent to which they do that in a way that resembles the genetic perturbations that you see in the cancer genome. Does that make sense? So quickly, imagine 4 DNA tumor viruses, clone all the genes for all the proteins, and do 3 types of assays to try to see globally where these viral proteins are actually touching the host network, derive a network model out of that, and we had then out of that a list of about 950 or so potential viral targets. And here's the last piece of data that I want to show you. This is a reference of cancer genes that are very well accepted, very well demonstrated. This is how typical cancer genome analysis overlap on top of the gold standard, which tells you that there's still many genes to be found, right? The viral targets that we found overlap approximately to the same extent as classical cancer genome sequencing, but they overlap in a very different way. And then look at this overlap over here, which is outside of the gold standard. Those are new potential cancer genes. And now they've been found using cancer genome sequencing and viral targeting. And so we think that that lists like this can serve as entry points for functional investigations of candidate cancer genes. I'll skip this slide because I think I'm going a little bit over time, and I'll just summarize by telling you that we're in the business of mapping the interactome network for human. We collaborate with people who are interested in graph theory to try to extract properties of these networks, and that has helped us over the years to help finding new cancer genes, contextualizing the disease, and then functionalizing as I just showed you in the last example. So I'll put my acknowledgement slide for maybe a question if I have time, if we have time or not. Thank you.